This is a production of Cornell University.
Today about diversity between species, and I might echo some of the points that Avi made earlier today. I was finding that things were not working exactly the same in Arabidopsis, or perhaps not as well in Arabidopsis as they did in tomato, hinting that perhaps different species do things a little bit differently. And so this subtext to this talk is going to be sort of whatever you thought was happening in one system may or may not be happening in something else. So I'll be talking specifically about abscission. So I need to define that first. And that's the process of releasing parts from the parent plant, things falling off. So, oh dear, something's right. Yeah, so you're most familiar with this in the autumn. Leaves fall off trees, and petals abscise in the spring. Of course, dandelion fruits abscise pretty much all the time, including when you don't want them to. And the most famous story of abscission may be the apple falling off the tree onto Isaac Newton. So in general, the location of abscission is not random. It's not simple breaking. Abscission occurs at a very specific position, the abscission zone. The abscission zone, which is here from a diagram of the abscission zone at the base of a leaf where it attaches to the stem. It's this little line of very small cells. And it can form in different positions in different plants. So it can be the base of the leaf, the base of the petals, below the fruit, below the seed. So it's something that happens in a lot of parts of the plant. Today, I'm going to be focusing particularly on abscission below the fruit. The most familiar abscission zones below the fruit are in the domesticated cereals. So you know this if you've studied domestication at all. In the cereal crops, such as rice and wheat and all the grasses, abscission and shedding of seeds is called shattering. So this wild wheat on the left shatters, that is, it breaks up. There's abscission in very specific parts of the inflorescence. Whereas the process of domestication is a loss of shattering, which was selected unconsciously by humans. And as if you're going out and selecting, as this guy is here, harvesting with a scythe, and then as these women are doing here, wrapping the stalks into sheaves, obviously what you're doing is you're selecting for plants in which there's no abscission. That is, the seeds are on the plant. Any seeds that are falling off end up in the mud and you don't collect them. And so you're automatically selecting for the loss of shattering. It probably took quite a while for this set of mutations to get fixed. But basically what it means is that all the domesticated cereals are natural mutants in which shattering is lost. So mutations that disable the abscission zone. And a lot of what we know about abscission zones come from these wild domesticated comparisons. And I'll be referring to a number of these through the talk. The other reason why this is important is that there's a lot of focus on improving novel crops that also need to be selected to reduce or completely eliminate shattering. So I've just put two here that we're working on right now. I'm not going to be talking about this work in this talk particularly, but Fonio is a species of Digitaria, which is edible, but and is grown as a crop in some parts of Africa. Teff is probably more widely cultivated. Both of those drop their seeds, although Teff sort of holds onto them a little better than Fonio does. So that if you're selecting for yield in these crops, if you want to do that to improve them, you've got to reduce shattering first, because otherwise, if you lose the seeds, then it doesn't matter how wonderfully nutritious or large that seed is. It's just sitting there in the mud and you're not going to get it. So it's important target for selection and crop improvement. Just a little bit of introduction to the grass family. I know it's familiar to many of you here, but just to remind you, it is a large plant family. It's about 12,000 species, so about 700 or more than 700 genera. Then I've just put 4 of them across the bottom. Of course, the big 3 are rice, wheat, and maize. I put sorghum and millet here. And I need to introduce you to just a little bit of grass floral anatomy, not much, but some. The grass flowers are arranged in little spikes, so these little structures known as spikelets. So this is a spikelet right here. And it's a series of bracts surrounding a fairly conventional flower. And the outer bracts subtend the spike. So there are 2 outer bracts, and then 1 flower, 2 flowers, 3 flower, 4, and so forth. These outer bracts are known as glumes, which is, of course, when people study grasses, they get depressed about it, and people call it the gloomy flora. But so 2 glumes are subtending the spikelet, and that's the bit of morphology I'll need to refer to for this talk. So here's another picture. This is switchgrass. There's first glume, second glume, and then the flowers are above that. So the glumes sort of demarcate the bottom of the spikelet. In rice, the abscission zone forms above the glumes. So the glumes are these tiny little structures here. The red dotted line marks the abscission zone. And then when the spikelet breaks, when abscission occurs, the glumes stay on the plant, these 2 little flaps, and then the rest of the flower and the seed and everything falls off. So you can show that in this little diagram where I made the glumes in blue and the abscission zone is red. Then there are a couple of sterile flowers, and the big fertile flower is shown as this big green egg-like thing. If you look at something like Brachypodium, there's also abscission above the glumes. So there's the glumes at the bottom, these little blue things here. And then you've got multiple flowers in Brachypodium, and there's an abscission zone forms between each set of flowers, but still above the glumes. If we look at something like millet or panicum, like switchgrass, on the other hand, the abscission is below the glume. So there's a glume, and the thing breaks off, the spikelet falls off entire. So there are no bracts left on the plant. So you've got a couple of blue glumes here, and then an abscission zone right below it. So in some things that I've just showed you, you've got abscission above the glumes, and some it's below the glumes. So how does that sort out across the grass family? Well, Yunqing Yu, who's a postdoc in my lab, just characterized a whole bunch of grasses based on stuff from the literature and her own observations, and then mapped the position of the abscission zone onto the grass phylogeny. And so blue is the abscission above the glumes. That's like rice and Brachypodium, which I just showed you. Here's rice, here's Brachypodium. And that seems to be the ancestral condition. And then these things where there's abscission below the glumes, like Setaria, is shown here in green, and that's a derived condition. And then there are a few things that you're familiar with, like teosinte or wheat, in which there's abscission actually in the stalk of the plant. That's shown here on red. But for most of what I'm going to be talking about today, we're just looking at this simple transition between below the glumes and above the glumes. So that's the ancestral condition there. So this raises a really obvious question from the point of view of genetics and development. And the question is there just a single abscission zone module that the plant sort of just moves around depending on where it's going to form? There's a single module conserved, and then maybe the transcription factor or something that sort of says, oh, I'll put it here or put it there. That this would be expected. This would be one possible explanation of the evolution. And so this is the question I'm going to be talking about today. First, starting with simply histology, that is cutting sections right through the the abscission zone to see what it looks like. Moving on to QTL studies and genome-wide association studies, and to RNA-seq, trying to address this relatively straightforward question. So let's start with the histology. Most of what we know comes from rice. There's been a lot of work on abscission, that is shattering, in rice, comparing cultivated rice with its wild shattering relatives. And certainly in Asia, the wild rice, which shatters, is a real problem in agriculture, because obviously then it's dropping seeds to produce weeds and get in the field and compete with the crop. So if we take a vertical section right down through here, so a longitudinal section, we just slice it down through and stain it, what you get is something that looks like this. And this is, we've done this in my lab. So these pictures are from my lab, but they're pictures in the literature that are very, very similar. So the abscission zone, the place where things actually break, is this little line of cells right here. And this, I need to introduce you to a few histological stains so you know what the colors mean. So this is stained with saffron and O and fast green, and saffron and O stains both lignin and suberin. So the pink indicates lignin. And then the abscission zone itself is not lignified. So this is the abscission zone here, both sides, and this is a vascular strand going down, connecting the bottom of the spikelet down to the rest of the stalk. So lignin and no lignin. This is another stain, fluoroglucinol HCl, which is much more specific for just lignin. You get basically the same pattern. That the place where it actually breaks is this zone of small, thin-walled cells. So that's fine. It's kind of intuitive. If you have trouble seeing it, I've just drawn a squiggly little line, and the squiggles mean nothing other than the fact that I can't draw very well on a mouse pad. And people have looked at this. This is one paper from the literature looking at the African rice compared to a mutant. Here's, obviously, we've now got acridine orange, which is also still staining lignin. It's just now green. There's the abscission zone. And in one of the non-shattering mutants, in the domesticate, lignin now extends all the way across the abscission zone. So it just doesn't have that break point. So that suggests then that a functional abscission zone requires a narrow layer of small cells and differential lignification. And this is sort of, if you look up abscission zone in the literature, this is sort of the description you will get, is that you've got to have different lignification and then this narrow layer of small cells defining the breakpoint. So that suggested a fairly straightforward set of experiments, which turned out to be really good for summer undergraduate students. I highly recommend anatomy and histology if you've got summer interns, because they get to look at something nobody else has ever looked at before and make a discovery and produce some pretty interesting data. So Rachel Tavares, who was an undergrad intern, now a grad student at UMass, looked at Brachypodium. And this is abscission, we call above the glumes. So it's right here. We expected it was going to be just like rice. We expected we'd have this same rice type abscission zone in between the flowers and above the glumes. And so Rachel looked at Brachypodium distachyon, and what she discovered indeed was this nice little line of small cells right here, very clear. But if you look at the staining, this is saffron and O, so the pink means lignin. What you discover is that the pattern is exactly backwards of what I just showed you in rice. So here's the rice, lignin, and then the abscission zone itself is non-lignified. And in Brachypodium, the abscission zone itself is heavily lignified and not on either side. Here's this other, more specific lignin stain. So that was distressing. But we thought, oh, well, there's still a nice little line of thin-walled cells. So what about abscission below the glumes? what does that look like? Well, this was studied by John Hodge, who was a master's student in the lab. He's now a PhD candidate at Oklahoma State. And John got looking at the abscission zone in Setaria viridis, which is closely related to switchgrass, and Phonia. And he's looking at this region right at the bottom. The abscission zone is right here, right below. So it's sectioning right through here. And I put this embryo over here. This is a mature grass embryo just to prove to you that this spikelet really is mature. It's not that we got the stage wrong. There's basically no little line of cells at all. We've looked at enough of these. I put this dotted line on just to show you where it breaks. We've looked at enough to know that's where it happens, but you're not seeing any clearly differentiated abscission zone at all. And if you stain it with these, like saffron and no, here's the lignin. It's just around the outside. And I put Brachypodium next to it just so you can see the comparison. There's the Brachypodium abscission zone, which remember is the inverse of the rice. And even if you stain it with agridine orange, it's not just the stain, there's the lignin out here, and there's lignin, of course, in the vasculature, but there is, this thing breaks right here. So there is no cellular differentiation. This is stained with DAPI, which lets you know where the nuclei are and points out live cells. The cells are alive right through there, so nothing's dying. And I don't want to spend too much time on this one point, but we've done a number of other approaches. We still can't figure out what it is that marks the abscission zone in this particular species. So just to summarize the histological comparisons among these three species, in rice and Brachypodium, yeah, we've got this canonical abscission zone where the abscission zone cells are significantly smaller than the surrounding cells. Setaria, once you know where to look, they are slightly smaller, but not compellingly so. Differential lignification definitely in rice and brachy, but not Setaria. And the abscission zone itself not lignified in rice and Setaria, but not Brachypodium. So we're not seeing a lot of conservation. So we then tried to rescue that with another hypothesis and said, well, maybe, you know, each one of these things is in a different clade, and at least it's representative of the clade, so that everything that's related to Setaria will have a Setaria abscission zone and Brachypodium, so forth. And so our undergraduate intern last summer was Poaceae leva, who decided to get serious about this. And we said, fine, just section everything you can get hold of. Well, it turned out everything she could get a hold of was, and she could get through in a short period of time, was an enormous amount. So it's all these species that are up here that she's actually looked at. And I'm not going to walk you through every single species, but there they are. They're just all different. You know, this one does, there's the abscission zone. It's not lignified at all, either side. And that's Brachypodium, which you've already seen. This is Tef over here, and Tef actually breaks right there. So yeah, they're all different. And to figure out whether that tracked the phylogeny, she mapped all these characters onto a pruned grass phylogeny. And if you just look at the position, that's coded as, you know, basically 1 is above the glumes and 2 is below the glumes. And that isn't consistent within any, you know, it doesn't match the phylogeny. In terms of having the tiny little cells, 1 is having small cells and 2 is not having a set of small cells. That doesn't match. And D is the cell wall pattern. So green means not lignified, and pink means is the color of the stain, meaning lignin. And that doesn't track the phylogeny at all. So in terms of the question we're trying to answer, if we look at histology, the answer is no. There is no conservation in the structure of the abscission zone. So that was not great, but you start thinking, well, maybe there's some underlying genetic conservation, even though there's no histological conservation and RNA-seq. So let's look at QTL studies. And certainly there have been a lot of them, particularly in rice, as I mentioned. They've identified several genes that control the abscission zone. And a few years ago, our colleague Andrew Doust wrote a review paper in the American Journal of Botany where he took the rice chromosomes and put on them just the location of the QTL from all the published studies up to that point. So QTL are here in these black boxes. And the genes are listed under here. We'll not be mentioning too many of the gene names today, but they've been well localized and some of the genes have been cloned. And the question is then, what does it look like when you put other species on here? So red is Zea, green is sorghum, brown is Setaria. And what you see just by looking at how the colors line up is, in fact, there's not much overlap except in this one region here. So a lot of stuff that seems to be unique to rice, a lot of stuff that's unique to the other species, possibly some similarity. And that region of similarities seems to all cover this one YABI transcription factor, which is known as shattering 1, which was originally identified in a sorghum cross, as shown here. So I'll be focusing on only a couple of genes today. So this is one, if you can hang on to this name, the YABI gene of shattering 1, that is shared across a lot of different species in these QTL studies. So that raises a question about natural variation. Remember, I started out by saying our ancestors who domesticated all the cereals were selecting unconsciously from standing variation in wild plants. And if that was going to work, I mean, we know it did work, there have to be non-shattering mutations that are relatively common in wild populations. If the mutations aren't there, then it doesn't matter how much you select on them, you don't get anywhere with it. But nobody's actually surveyed a very large set of wild germplasm for reduced shattering and then gone on to clone a gene. So I'm going to tell you about doing that now, because we've did that. And the plant that we looked at was green millet, which is Setaria viridis. And it's shown here growing in its natural habitat in the United States, which is right next to a highway. You can see the tarmac over there. It's a very weedy plant. And we collected it pretty widely throughout the US and got hold of some material outside of the US. It's a small diploid plant. It's been being used as a model for a number of things, particularly for studies of C4 photosynthesis, because it's small, it's diploid, it's rapid cycling, it can be transformed. So quite a few labs are using this as a model. And quite recently, we've been involved, I should say, is actually folks at JGI, so this group led by Jeremy Schmutz, have produced a new assembly of a reference line. So there was an old version, and this is an updated, much better version, line known as A10, which I may mention during this talk a bit. I'll try not to. But, so it's a really nice assembly. It's basically all 9 chromosomes and relatively small number of contigs with a real good N50. So we have this really lovely assembly. And as part of that project, we also collected and sequenced 598 diverse lines particularly from the US and Canada, as well as elsewhere. And I'm putting this map up here for a couple of reasons. One is, when you're working on natural diversity, in most cases, you don't have a gene bank to go to. If you want to you go get it. So that's what we did. The yellow dots are trips that, well, I did that one. The people from my lab made over a period of several years. Max Feldman from Ivan Baxter's lab did all the blue loops, and then green and red was from Pu Huang. This map also shows something else, and that is, of course, that St. Louis is the center of the universe. And also that Setaria viridis is very weedy and grows really well along interstate highways. One of the people formerly from my lab, I do need to give a shout out to, is a guy named Matt Estep, who's now at Appalachian State. And he did the collecting all the way up to here. And there is another one of the reference lines that I'll probably mention at least once, called ME034, that is very widely used now. And the ME stands for Matt Estep. So so his line is becoming very widely used. And so the JGI sequenced libraries for each of these things, covered to very high coverage. Each one has been assembled de novo. And we've got a lot of SNPs, did a lot of presence-absence variation. 98% of the genes are shared among all the species. And we've done a bunch of, Adam Healy and Sujan Mamidi at the JGI Hudson Alpha have done a lot of population genetics on this, which I'm only just going to mention more in passing for this talk, but there's a lot there. Population genetic analysis shows 4 major introductions from the old world. So there's the stuff in pink we're calling the central population, and that is genetically very similar to material from China. The blue is very similar to things from Southern Europe and Turkey, sort of Caucasus. Green, there's the northern population, which also extends across Eurasia. And then there's the orange and the West Coast population, which is also parts of the Western Asia. So a lot of introductions from the old world. And Pu Huang, who's now at BASF, phenotyped a subset of these lines, about 200 of them, and assessed them for shattering. So here's a bunch of plants over here, and you can see there's a big pile of seeds, spikelets, that have fallen off. Obviously a very high shattering pile of plants. And from that, he did a genome-wide association analysis, which identified a single peak right here, covering a pretty large QTL, but it didn't cover any previously known shattering locus. So none of the stuff from rice. What he did find was he managed by various combinations of tests was a causal gene, which we're calling less shattering 1, just because it reduces shattering. It doesn't completely abolish it. It's an uncharacterized MIB. So there's a high shattering line, which happens to be like ME34, and there's a low shattering line, which is the reference, in which there's a point mutation that causes a non-conservative amino acid substitution. So to validate that, this was knocked out with CRISPR. So here's the wild type allele over here, free shattering, and then the CRISPR line is substantially less shattering. You know, once again, it's not a complete abolishment of shattering, but it's a lot less. And we validated that by measuring the strength of the abscission zone, which is, that's a technique that's used frequently in this approach, where you take the inflorescence and hang it upside down on a force gauge, and then grab one spikelet with a pair of tweezers and pull. And the force required, breaking force required for the abscission zone turns up on the force gauge. This is a very boring experiment, but it gives you pretty good data, which is shown here. So here's the tensile strength of the abscission zone. The high shattering wild type, this is ME34, is shown in red, and you can see after about 18 days after heading, it's dropped close to 0. The slightly lower shattering wild allele does shatter, but later. And then the CRISPR line the tensile strength stays very high. So changeover development validated that. And then we also found that the domesticated version of green millet, which is foxtail millet, so this is the wild one, this is the domesticate, actually has a transposon in LES1 that appears to disable it. So it seems to be a gene that has contributed to domestication. Obviously can't be the whole story because it's not a complete non-shattering phenotype. So that lets us then go back to some QTL studies that have been done by colleagues and also published. And the question is, does it fall under any known QTL for shattering? That is it a domestication gene? So here's a QTL map of shattering from an RI line that was made in, Andrew Doust made and has developed in his lab between a wild Setaria viridis and the cultivated foxtail millet. SH1, which I mentioned to you earlier, the one that comes up in all the other QTL studies, is right there on chromosome 9. And the question is, does LES1 fall under this chromosome 5? And no, it doesn't. That's not LES1. LES1 is actually over here. So what we're thinking right now is that the transposon in LES1 probably reduced shattering and may have been involved in early domestication, but then subsequent much stronger mutations were involved in actually producing this completely non-shattering domesticate. So just to summarize the Setaria genome-wide association studies, we've got a high quality genome, a lot of wild diversity, a lot of good population genetics, and then use GWAS from that to find a novel domestication locus, which was validated with CRISPR, and obviously produces a resource for exploring other traits. What this does is it adds a species-specific gene to the list of genes from other species in other QTL studies. So we've just talked about LES1, and these other ones, again, I'm not going to go through them in detail, but what you'll see is that they're each one, this is a rice one. This seems to be specifically rice. This is rice. These guys are Triticeae only. That's wheat and barley. And only SH1 seems to be in multiple species. So what we're seeing is that from these QTL and GWAS studies, the controls of abscission appear to vary among species. However, this kind of work is not designed to get you the whole network. And so it's quite possible that there is indeed a conserved network that's moved around the plant, which is where we started, of course, but we've just found a whole lot of different ways to break it. And so we say, is LES1 involved in shattering in other species? And we'll come back to that in a minute. So what we've shown is based on getting back to our original question, from the question of the histology, there's no conservation. From QTL and GWAS, it's inconclusive, maybe shattering one NABI. So obviously, what's the thing to do? The thing is to do RNA-seq. And so this was a collaboration led by Yunxing Hu in my lab and with Hao Hu and Andrew Doust at Oklahoma State. And remember that we have rice breaking above the glumes, Brachypodium between the flowers, and Setaria, the abscission zone, below the glumes. So what Yunxing did was dissected out the region of the abscission zone for each of those 3 species. So you've seen all of these so far. So here's the abscission zone. She dissected out the regions roughly with this orange box. And then as a sort of internal control, she looked at the area right below the lower region and the area right above, that is the upper region. And so could compare the genes that were expressed in the abscission zone to the ones above and below. And this Venn diagram simply shows that the set of one-to-one orthologs is largely the same among all species. So that the set of genes that are expressed in that region is shared. So then the question is, which ones of those are specifically restricted to the abscission zone? And she did a bunch of different analyses. Yunqing and Hao did a lot. I'm just showing you the results of WGCNA, and just to walk you through this diagram here. What we have is a heat map of expression levels. These are 3 replicates for below the glumes, 3 replicates within the abscission zone, and 3 replicates above the abscission zone. This is at a young developmental stage. And then the same set of replicates below the abscission zone and above the abscission zone at an old developmental stage. And for this particular cluster in rice, there are about 3,000 genes that are clearly distinguishing the young stage from the old stage. They're up at the young stage, they're down at the old stage. And each species has modules that look like that, and they overlap significantly. So there's the 3,000 ones I just showed you in rice. There are about 2,200 in Brachypodium, and the overlap among those is about 850 genes that are differentially expressed, shared between them. Highly significant overlap, suggesting there's a lot of similarity in terms of the developmental stages. But of course, what we want to know is what's happening with the abscission zone itself. So here's a rice set of genes that are highly expressed in the abscission zone at the young developmental stage, much higher in the abscission zone than either below or above. And again, every species has a couple of abscission zone specific modules at particular developmental stages. So the question then that Yunqing and Hao asked was, to what's shared among these? Here's rice, there's a set of 962, here's Brachypodium, there's 111. What is shared between those modules? And the answer is basically nothing. There's a little bit of overlap, which is barely statistically significant, given the number of comparisons. Very low numbers. And of course, what we really want is the 3-way overlap. And there are 3 genes. So there's a MIB, there's a lysine decarboxylase, about which we know nothing, and coumarate ligase, which is presumably involved in lignin formation. So our first thought was, well, maybe we just messed up the analysis. Maybe we should be considering analyzing things slightly differently in terms of which genes we included and how we did the comparison. We did a completely different analysis, and we got exactly the same result. In fact, the second analysis, we only got 2 things overlapping, although they were 2 of the same ones we saw in the first one. So here's the MIB, highly expressed in the abscission zone at a young stage, and the lysine decarboxylase also among all 3 species. There's the expression domain of this MIB is in the abscission zone, so that's good. In 2 of the species, couldn't pick it up in Setaria, which either means that the RNA-seq is misleading or this method in situ is not sensitive enough. You might be thinking, well, what happened to LES1? And the answer is it wasn't in this analysis at all because it was not differentially expressed in anything. Not in Setaria viridis, very low level of expression in rice, and not expressed at all in Brachypodium. So again, it's not only species specific, whatever it's doing is probably not specific to the abscission zone. And the effect on shattering may be a byproduct of something else. And if we go back to the known genes, the genes that have been known in rice, these are ones that have been identified in the literature, and they're abscission zone specific in rice. But if you look at those same genes in the abscission modules as to where they fall in the other species, like this. Again, they're not always even in an abscission module in the other species. Thinking that perhaps we needed to look earlier in development, and Yunqing did a lot of in situ hybridization at earlier stages. So this oldest stage of in situ hybridization was the youngest stage from the RNA-seq. You can see very clearly gene expression. This happens to be apetala 2. And you can see it at the earlier stage. So this is rice. Again, this is confirming published data. And then if we look at the other 2 species, there's no expression there. Let me put that's where the abscission zone is. And what you can see is that they're really just, it's not, simply not expressed in the abscission zone in the other species. Same thing for other things. So if we get back then to the question that we started with, is the abscission zone conserved by a developmental, controlled by a conserved developmental module? The answer is histologically no. The QTL is inconclusive, maybe SH1. And RNA-seq points to only 2 genes. Looking at SH1, it's mutated in many domesticated cereals. We don't know, as with everything else, we don't know what any of these things do. Here's SH1 in situs. I think I'm going to skip that, but the gene networks differ among species. So although it may be a conserved component of the network, its specific role may be differing between species. So the overall answer to the question is no. The set of genes involved in an abscission zone is largely shared, but how they are deployed is mostly different. The practical implications of this are mean that for improving the orphan crops, remember I talked about Taphonia right at the beginning, or novel crops, things like perennial wheatgrass. For any of that improvement cannot proceed without reducing shattering. If these networks are species specific, we need to study each crop individually as to how it actually is put together. SH1 MIB26 may be exceptions, and we're certainly pursuing looking at those to see how they are deployed, particularly in Taphonia. And the other thing that we're pursuing is exactly how they work, because that at the cell level is not known. In general, we're thinking that most of the genes that are necessary for abscission are probably establishing development of the spikelet and the pedicel rather than controlling abscission per se. That is, if you affect spikelet development or pedicel development, it's going to have an effect on the abscission zone, sort of indirectly. And the abscission zone itself may be controlled by only a handful of genes, or may be deployed only at the very last minute before abscission, or it may not be transcriptionally regulated. So with that, I'm going to stop and thank the team, particularly at Hudson Alpha, the people who did a lot of the work on the Setaria genome and all the diversity work and the GWAS studies. It's a great bunch of people to work with. And then this is my lab at the Danforth Plant Science Center. Most of what I talked about today was work of Yunqing Yu, and yes, that is the Stanley Cup. So I'd like to thank you for all your attention, and thanks again to the organizers for putting together a great symposium. So.
Thank you so much, Toby. That was a really interesting talk, and I see we already have a few questions in the chat. It looks like the first one was from Hannah Jeffrey. Do you want to unmute yourself and ask that one?
Yes, thank you. I was interested in that map that you had shown earlier, and I realized that it may be a basic question from some perspectives, but I was thinking about it in terms of ecology. Just I don't know.
Well, I think Hannah just disappeared.
I'm fine.
Okay, good.
It's very loud where I am. I was thinking about it from an ecological point of view. And I was wondering, do you think that maybe you could find more homologs to important genes near the Illinois area where a lot of the species seem to radiate out from?
It's possible. Well, we included, I mean, every one of them has been sequenced in detail. So we've certainly looked at the whole array. Because there's not only been a lot of the introductions seem to be in that part of the Midwest, but there's also been a lot of admixture in there. And so that could be an interesting place to look at more closely, and specifically look at the admixed lines in particular.
Well, that is very cool to see that. Perhaps there would be something unique that would have popped up later on, as opposed to closer to the epicenter.
Yeah, well, I think the one thing is to keep in mind is that the epicenter is partly driven by the introduction history of this thing. And we don't have a real good picture of the diversification across Eurasia. The sampling is just super sparse there. And I think your question probably would be really interesting to address closer to where the species is actually native. And that may be where you actually see a better signature of the ecological interactions. But yeah, it's a really good question.
I agree. Thank you.
Thanks, Hannah.
No problem.
Looks like we have another question from LN.
Elizabeth.
Hi.
Hi, do you plan to automate your tensile experiments in the future? Because that's really cool work, but someone pulling it by hand may affect the quality of results.
Yes, it also affects the enthusiasm of the person doing the pulling. It's not a great experiment. We've tried other assays. We have other ways of estimating kind of how many seeds fall off, counting seeds or weighing them and that sort of thing. The tensile strength is very nice and quantitative. And yeah, if we had a way to do that more easily, it certainly would be good. But I haven't been able to come up with the challenging part is simply getting hold of the spikelet. And if there was a way to grab it in some good way, it would be great. So yeah, if you have any ideas, I'd be happy to hear them.
Thank you.
Thanks.
And then it looks like there's a question from Stephen Merrick.
Yes, I was wondering about if you looked at any of the microRNAs or small RNAs in the abscission zone region, and if they have any homology to any of the identified differentially expressed genes.
Yeah, no, that's sorry, what? Say that again.
Or their UTRs.
Yeah. So that's a really good question, and it's something we've thought about and just haven't made the libraries to do the sequencing. But it's as we're going forward, we are almost certainly going to be doing that. Certainly the A-petal-2 genes, which do come up in some of these species, are regulated by microRNAs. So yeah, I guess the answer to your question is no, we haven't done it, but yes, we'd love to and will be.
Thanks.
Yeah.
And we also have a question from Jim Holland.
Oh, hi, Toby. That was a great talk. I was wondering about looking at genera where there's variation within the genus for where the shattering occurs. And my recollection was in Avena that different species will shatter at different places in the floral structure. So I'm wondering if you've looked at cases like that.
We have not, certainly with Avena, that you're right, the different, different cultivated lines and different species do break up in different ways. And that's also true with the wheat wheat and barley relatives. They just break in different places. I think we had gone into this assuming there was just going to be a completely conserved network, and so we hadn't really dug into the within species diversity. But given what we know now, that might definitely be worth pursuing.
Jeff Doyle, you want to go ahead and ask your question?
Yeah, Toby, nice talk. Is cell death involved in abscission?
You know, the literature is ambiguous. And if it is involved, it's pretty late. As some species, and I think there's work in lychee where abscission of one cell layer from another involves cell death, but in the cereal crops, the assumption, which I think hasn't been very well tested, the assumption is that it's an active process. That is, the cells right there at the abscission zone actually secrete cellulases and pectinases out to dissolve the cell wall, and they can't obviously do that unless they're alive. So it may not, cell death may not be involved. The exception may be in teff. Is the place where teff breaks, it actually looks like those cells just fall apart. It becomes necrotic or something. So yeah, I guess the answer to your question is we don't know.
Yeah, I guess I was just thinking that maybe that would be a process that would be, sort of difficult to detect by any of the histological methods or RNA-seq, but could account for some program in there that would actually cause a group of cells to die and create that break point.
Yeah, and that may actually be a good possibility. And one of the challenges of this is once you start focusing on things like the actual breaking process itself, it gets hard to study because the things break. So that you can't, things just fall apart on you as you're trying to work with them. But yeah, no, I think that's a really, really good possibility.
Thanks.
Yeah.
And we have also a question from Chelsea Specht.
Yeah, Toby, thank you. That was a fantastic talk. I was also thinking like Jeff about programmed cell death, and also going back in my head as what are the processes to study that. I know a lot of it was with nectaries where things start to degrade and then you get the secretion. But with a abscission zone, it's different in that you don't get that cellular secretion along with the death. You actually get, I don't know, like you were just saying, the cells kind of break apart or they lose their cell walls, right? They kind of dry up, I guess, for lack of a better word. But I was thinking about the signaling, if there's any way to detect some kind of, I don't know if it would be a signaling or hormone or some sort of small molecule that might be involved with setting up a particular zone where the cells then are no longer involved in growth, but rather somehow are enzymatically broken apart.
Yeah, I mean, I think there's got to be something like that. We're currently focusing on a couple of things. One, because we actually have a CRISPR mutant for SH1 that Yunqing can compare between wild type and mutant. And we're focusing on cell wall structure. So like the distribution and possible breakdown of the various carbohydrates, the pectins and the xyloglucans. And also there seems to be a hormonal response involved. So yeah, I mean, it's kind of watch this space. I don't have, the data are too preliminary to talk about, but yes, I think the situation you just outlined may well be what's going on.
Well, it's really interesting, and I really appreciate your real rigor and efforts in looking across all these grasses and finding that every time you look at it, it's different. And while that's that's challenging, it's also incredibly exciting to think about all the ways that grasses have come to the same conclusion, but doing it differently to affect, thinking about ecologically what differences that might make, or what these responses to be, and what adaptation these plants might be using this differential response to evolve towards is really interesting as well.
Yeah, from an evolutionary point of view, it actually makes no sense, because all of these things have abscission zones. They're all, if you use a strictly sort of phylogenetic definition of homology, they're homologous in the sense that they all go back, everybody has an abscission zone. So if you've got a perfectly good one, why move it?
Yeah.
But that's where it is.
Thank you.
Thanks, Chelsea.
It looks like, Jeff, you have another question?
Yes, thanks. Yeah, just it occurred to me, when you were showing all those really nice histological pictures about how variable different species were, it makes me wonder, do you see that same, a lot of that same variation perhaps within a species? Did your student look within a species to see how variable that was at that level?
No, we haven't. And that sort of follows, Jim Holland's suggestion is there are species that do vary. Mm-hmm. And we just haven't drilled down to that level of variation. But yeah, I think that starts to be, it wouldn't have occurred to me that genetically or histologically there would be variation within a species for abscission, mm-hmm. even though we know the position of it can change. But now that we're finding this amount of diversity, absolutely, I think that would be one way to go, would be to look within a species.
Yeah, I wasn't thinking so much of position as Jim questioned, but actually lignification and, like all the whole suite of characters, the different types that you referred to essentially,
yeah.
Whether that's actually something variable.
Yeah, no, that's a good question. And we don't know. We don't have the data.
All right, thanks.
Yeah.
This has been a production of Cornell University, on the web at cornell.edu.